In this video, we focus on factors that affect the ability of enzymes to carry out their function as catalysts of chemical reactions in the body. We will here focus on two main factors that influence enzyme function and talk about what role each of those play and how well or how poorly enzymes function. Those two factors are temperature as well as pH. And temperature is the reason why I have a picture of a feverish child here as we lead in. What we will see is that temperature has a dramatic impact on the function of enzymes. And when the body's temperature isn't properly regulated, meaning it's too hot or too cold, enzymes are not going to function optimally. And that leads to a variety of symptoms that we associate with fever or symptoms that we associate with such things as hypothermia if the body's temperature drops too low. Let's go ahead and take a closer look at these two factors, starting with temperature. In general, with enzymes, when we look at the relationship between the temperature of the system and enzyme function, what we see is a plot similar to what's on the screen here, where the rate of the chemical reaction increases as we increase the temperature of the system up to a point. And at that point, which we call the optimal rate point, the optimal temperature for that enzyme to function, which in the case of this system is somewhere between 40 and 50 degrees Celsius, after that point, the activity of the enzyme drops off dramatically, meaning that the ability of the enzyme to catalyze a reaction falls off like a brick here. And the reason for this relationship is when we look at this left-hand end of the spectrum, going up toward the optimal rate, with an increase in temperature, we are increasing the energy available in the system, and that's going to facilitate the enzyme in acting and catalyzing the chemical reaction. However, when we get to this point of the optimal rate and we go beyond that and we see that rapid decrease in enzyme activity, that is explained by denaturation of the enzyme, whereas the three-dimensional shape, that is the secondary, tertiary, and quaternary structure of the enzyme is disrupted, the enzyme no longer has the proper 3D shape features in order to properly catalyze chemical reactions. And denaturation is generally non-reversible. So once an enzyme has been denatured and is no longer active, it can't be cooled off typically and refold properly into the original three-dimensional shape. Instead, in the case of the human body would need to create more of that particular enzyme for the reaction to be restored. And this is one reason why having a fever makes a person feel kind of poor over the course of several days, is because of the fact that enzymes are damaged when the body is at a suboptimal temperature. Now let's consider the impact of pH, or acidity and basicity, on enzyme function. In order to do this, what we need to keep in mind about enzymes is that they are constructed from the 20 amino acids, where the R groups on the 20 amino acids that we learned earlier are going to impact the ability of the enzyme to bind to its substrate and also the ability of the enzyme to catalyze a reaction. Particularly important in the context of pH are the amino acid residues, the R groups, that we recognize as being acidic or basic. We can recognize those with the orange circles on this chart and also with the blue circles representing the basic residues. In cases where these particular amino acids are present at the active site of the enzyme and responsible for binding to the substrate, if the environment of the enzyme is too acidic or too basic, the protonation state of these particular residues will be impacted, meaning that they will either lose or gain a proton depending upon whether they are acidic or basic. The consequence of that is if the pH is not correct for the particular enzyme, the enzyme won't be able to properly bind to its substrate any longer. The other way that pH impacts enzyme function is that in the case of some enzymes use these amino acids to act as proton donors or proton acceptors to catalyze the chemical reaction by transferring or facilitating the transfer, I should say, of a proton onto a substrate. That can be an essential step in an overall chemical reaction process. And if the R group is not properly protonated or deprotonated for that to happen, then the reaction can't be catalyzed properly. Hence, pH is going to play a critical role in determining the functionality of a particular enzyme, where if the pH is not correct for that particular enzyme, then the reaction is not going to take place properly. Different enzymes are optimized to work in different environments, and based on those different environments, their pH requirements will differ. For example, enzymes present in the stomach are optimized to work at a very low acidic pH, where enzymes optimized to work in blood work optimally at a more neutral pH. Similarly, when we think back to the role of temperature in enzyme function, enzymes are optimized to function generally in the environment in which they are typically found. For example, bacteria in hydrothermal vents have enzymes that are optimized to function at very high temperatures, whereas enzymes in the human body are generally optimized to function at 37 degrees Celsius or 98.6 degrees Fahrenheit, depending on what units you like to use. So in other words, the take-home bottom line message of this all is that enzymes are relatively picky creatures, requiring a narrow range of pH and a relatively narrow range of temperature in order to have optimal function. And if the enzyme is placed outside of this range, it is not going to be as effective at catalyzing chemical reactions. And as a consequence of that, ultimately, if we're talking about human health, health will suffer.